Okay, so next week's going to be a little bit different in that we're going to go over 2 chapters from the textbook. Chapter 2 is fairly short, it's about 11 pages. We're going to go through it in its entirety between this lecture and then our time on Monday. So as you prepare for our class on Monday, keep in mind that we're going to go a little bit deeper. So we're going to not spend a great deal of time covering what I cover in this lecture, but instead covering some other aspects and a little bit of some more concepts. Also remember on Monday that we have a few things that we need to finish up from today's lecture. We need to talk about gametogenesis, but let's focus on mutations. So a few key terms. First, Knudson's hypothesis, or Knudsen's hypothesis, is basically the idea that for many cancers, there are basically, in order to have a faulty proto-oncogene or a faulty tumor suppressor gene, you actually need 2 copies of the recessive allele. And so both of your copies need to be bad, because if you've got one functional copy, that tumor suppressor gene functions normally, oftentimes inactivating some cyclin-dependent kinase. And so his idea is that for many cancers, you would inherit one bad copy, one recessive copy of this from one of your parents, and then the other one would develop in that cell line or in some cell line during your life. Induced mutations, these are mutations that are caused by some external factor, UV radiation or alkylation or some carcinogen. And we'll talk about these in some more detail in a minute. Deamination. Deamination is one of the more common spontaneous mutations. And deamination happens when you remove a portion of a base. So you can remove a portion of cytosine and generate uracil, or you can remove a portion of adenine and generate a very strange base, and that appears very rarely, and it is called. Sorry, I lost it here. And so the deamination. Hypoxanthine is the base that is caused when you remove this portion of adenine. Depurination is the next most common spontaneous mutation. And basically what happens here is you keep that sugar phosphate backbone, but the purine, like guanine or adenine, is actually removed, leaving a gap in the DNA strand, preventing the DNA molecule from operating normally. Mutational hotspots, these are places in the genome where mutations become more common, and they're going to be in places in the genome typically where you have repeated regions, the same bases repeated over and over again. Also, you may have mutational hotspots in places where you have what are called palindromic sequences, where the 5' strand in both directions reads the same. Repeat mutations, these are mutations at mutational hotspots where you have a repeated region, and here the machinery that duplicates the DNA can make mistakes and duplicate the wrong number of copies, but most often these are caused by a uneven crossing over during meiosis, where these big chunks of repeated regions, as they form the holiday junctions and those chiasmata remain, when they are resolved by the machinery that cuts it in such a way that one of the homologous chromosomes, or at least one chromatid from it, gets less of those repeats, and the other one gets more of those repeats. And so through time, these repeat mutations can actually lead to large-scale changes, and we'll see that in a moment. Now, anticipation is the phenomenon that many inherited genetic illnesses become or appear at a younger age and increase in intensity each generation. And oftentimes these are genetic illnesses that are caused by repeat mutations. And so remember, every generation, the number of repeats can actually increase, again, due to this uneven crossing over. And so the genetic mutation, if it's caused by malformations in the protein because of the number of repeats, you end up creating too many of a particular amino acid. Let's say it's alanine. Instead of having 12 alanine residues, you may end up with 30 or 40 alanine residues. And over time, as that number increases, the functioning of that protein can become even worse. And so again, each generation, the severity tends to get worse, and the illness tends to show up earlier in the individual's life. These next two, non-homologous enjoining and homologous recombination repair, these are mechanisms that are used to fix double-stranded DNA breaks. Non-homologous enjoining, what the machinery does is it takes small fragments of double-stranded DNA and it attaches them at the end of where there's a double-stranded break, because you know that the ends of chromosomes have a longer 3' tail, and so the ends of a DNA chromosome are not supposed to be blunt. You're not supposed to have blunt ends, and so there are machinery that recognize those blunt ends and attach them together. And so the non-homologous enjoining can restore or fix those double-stranded breaks, but it can also lead to chromosomal mutations if the wrong fragment is attached to the wrong chromosomal blunt end. Homologous recombination repair is also another way in which you can fix double-stranded breaks, fix these blunt ends, and it requires reading the homologous sequence on either the sister chromatid if you're in G2, or reading the homologous chromosome and using that to actually write the portions of DNA that are missing. Again, is carried out by very specific machinery that are designed to recognize the issue in the DNA and then to repair it. All right, so here are our induced mutations. Up at the top, you can see UV exposition, exposure to UV radiation. The most common mutation that results from that is what's called forming a thymine dimer. And so it forms bonds between the carbon residues in those neighboring thymines. Now, this is something that can be repaired directly, and we'll see direct versus indirect repair a little bit later. But it is something that distorts the way DNA functions and distorts the way DNA is read. So it's not a mutation in the sense that we typically think of mutations, and that the sequence of the DNA is changed. It's just how you read the DNA is changed because of the inability to read these bases correctly. Alkylation is the attachment of a methyl group to the carbon, the oxygen attached to carbon 6. And so if we take a look here is carbon 6 on this guanine, and here's the oxygen attached to there, and then here's the methyl group that has been added to it during this alkylation. And so the main problem here is that this guanine, this alkylated guanine, no longer forms hydrogen bonds with cytosine. Instead, it'll form hydrogen bonds with thymine, leading to the wrong base being added to the complementary strand if this residue is on the template. Then the last one, reaction with a carcinogen. Reactions with a carcinogen tend to add weird molecules onto your DNA strands. And so here we formed what's called an adduct, where we added this really large complex molecule onto our guanine residue. And so here again it distorts which bases are going to form hydrogen bonds with this. So when this is on the template strand, it can lead to the incorrect base ending up on the complementary strand. And again, these are all examples of induced mutations. So they're a little bit different than the spontaneous mutations that happen as a result of an error in the DNA polymerization. These are caused by external forces and then might give rise to some spontaneous mutations. All right, so here's one of the most potentially devastating DNA mutations. This is called a frameshift mutation. And so in this specific example here, we've got a insertion of a G into the reading frame here. And so initially we've got okay, so here's the sequence here, and GCC, ATT, TCG, CCT, and so CCT. Oh, this was, I don't know why it's saying insertion here, because we're actually, oh yeah, we inserted a G and an A and a G. No, sorry, you inserted just an A and a G. Sorry, I had a hard time seeing exactly what was going on here. So we inserted an A and a G here, and so what it did was it shifted our reading frame. And so, because they're comparing them both to here. Here's the original sequence. So I was looking at this all wrong. So here's the original sequence here. Here's an insertion of a G. And then here is the loss of that particular sequence. But anyways, in a frameshift mutation, if you add or you subtract any number of bases that is not a multiple of 3, it shifts your reading frame. So each codon is 3 bases, and so if you add 1, it's going to shift your entire reading frame. If you add 2, it'll shift the reading frame. If you take away 1, if you take away 2, it's going to shift the reading frame, and that trickles down the entire length of the chromosome. So not only would that gene be altered in terms of the RNA product being translated differently, but any gene that comes downstream from this frameshift mutation is also going to be impacted by the mutation. All right, point mutations. So these are what are called single nucleotide polymorphisms. This is where you have basically a spontaneous error result in the wrong base being added, and there are multiple forms that this might take. One is a silent form, and that's where here we've switched the thymine to an adenine, but it resulted in the same amino acid being translated. So that codon is translated into the same amino acid. Here you have a nonsense mutation in which, let's say that this adenine was mutated into a uracil, and now instead of presenting ATC, which would give us, so here's the DNA mutation. Originally it was TTC, which ultimately would be translated into lysine. Now it read ATC, which the RNA version of that is UAC, and that is one of our stop codons. So what it does is it terminates the translation early, giving you a shorter polypeptide and a shorter protein. Usually non-functioning, because it usually can't fold correctly if you end up with a much smaller protein. Missense So missense is where you've mutated a section of this codon. It is translated into a different amino acid here. However, in conservative, you basically maintain the same overall chemistry. And so you see here lysine is a basic or has a basic R group, arginine has a basic R group, and then threonine on the other hand though has a polar R group, and so it's going to have a different chemistry. And so missense mutations are always going to result in a different amino acid appearing in your polypeptide, but if the amino acid behaves the same chemically, you can oftentimes generate essentially the same protein, even though the primary structure of that protein is going to be different. The overall function of that protein is going to be the same. All right, so slightly larger scale mutations, but not as large as what we're going to see in Chapter 3. These are, say, duplication events or inversion events, or in this particular example, what are called transpositions. And here you have an enzyme that will cut out this particular section called the transposase enzyme, and then you have that other machinery that will go and will actually physically cut this section of DNA out of this portion of the genome and then insert it at the target site. And so this functions nicely for facilitating varieties of particular gene expression, but if it inserts in the middle of a gene, it can drastically change how that protein product, the nature of that protein product. This can also, during this transposition, be added in backwards, which can change the way it's read and change the protein product. And these are larger scale mutations, a lot of which we're going to talk about more in our video for Wednesday and during our lecture on Wednesday. All right, so these, remember, we were talking about mutational hotspots are where you have palindromic sequences and when you have repeated sequences. And so here are 4 diseases: Huntington disease, Fragile X, myotonic D, and Kennedy, that are the result of a increase in the number of repeats. So here's the codon that in Huntington disease, CAG, a normal allele has anywhere from 11 to 34 repeats, and then the mutant allele has anywhere from 42 to 100 repeats. And this would be a good example of one that can facilitate anticipation, where each generation the disease increases in severity and actually shows up earlier in age because the total number of repeats can increase generation after generation after generation. All right, so protein products, basically we have 2 different types. Most often what we're going to see in our mutations is going to be a loss in function. And here you've got a normal protein product. If without the mutation then triggers a improper protein product to be produced, or that the protein just never even forms because the mutation is incredibly severe. Now, in most of these, in order to actually experience the pathology, the individual has to be homozygous recessive for that mutation. And so that they don't make any of that protein product, because usually as long as you make whatever that protein product is, you're going to be fine. Now, there are some cases in which that's not the case, because you may only make potentially half of the product, and that might not be enough. And this is called heterozygous insufficiency, when the loss of function mutation is demonstrated even in heterozygous individuals and not just in homozygous recessive individuals. Then you have some mutations that are gain of function, and so Huntington disease is an example of this. And in this case, and in many of these, your original protein isn't even known. We're making some new protein product as a result of mutations, and that protein product is interfering with normal cellular function and development. And so that oftentimes, again, there may not even be an original protein. There might not be a normal functioning gene. And instead, we've basically almost created a protein product as a result of a mutation in maybe a region where you didn't even have a gene. And so these are more rare. Most of our mutations are going to be loss of function. But it's important to think about these mutations and remind yourself that what's happening at the molecular level is that it is changing or eliminating a particular protein that the cell needs to function properly. All right, so repair mechanisms. We just talked a lot about how various mutations can and do happen. And as you can probably imagine, the eukaryotes have a number of mechanisms to recognize, reverse, or stop the, to recognize the mutation, reverse the mutation, or to fix the mutation, or to eliminate that cell from being able to divide. And we've been talking about that the last few class periods, but in here we're going to talk about first reversing the mutation and then repairing it. So in order to reverse a mutation, you have to do what's called a direct repair. And so this, you have to basically eliminate whatever was whatever mutation happened. And so a good example of this is the alkylation. Here we have an alkylated adenine base, and here we have an alkylated cytosine base, and then we've got enzymes that can actually remove that methyl group off of the base and return the base back to its normal form. And so this would be a direct repair. You had some kind of alkylation event that attached this methyl group, and now we have some enzymes that can remove that methyl group and eliminate that mutation. And again, remember the example we gave for this and how this can trigger issues is the guanine, when it's methylated at the oxygen attached to carbon 6, no longer forms hydrogen bonds with cytosine and instead forms hydrogen bonds with thymine, which results in an issue in the DNA replication. So this is rare to have mutations fixed this way. Most often mutations are going to be fixed doing what's called excision repair, in which the mutation is recognized, and then there are machinery that will go in and will remove that mutated portion. And so here you had right here we have a mutation that is recognized. Here's a damaged base. It's removed, and then there's another machinery, the AP endonuclease, that will cut out the area around that damage, and then either allow DNA polymerase to come in and just start writing, and then write all the way to the end, displacing the strand that for the most part was fine, and then at the end ligase will come in and seal that up, or it may cut out that section entirely to begin with, and then RNA polymerase will come in and write just to where there's already a base in place, and then ligase will come in and will fix that nick. Either way, again, this is the most common machinery for fixing mutations, at least for fixing mutations that aren't a double-stranded break. Recognize the error, you cut out bases on either side, and you write a new strand. And you might think that this seems pretty wasteful to cut out more than just the error nucleotide, but it uses machinery that is already active in the nucleus and already writing DNA. So it actually creates a high level of efficiency and allows the machinery that is already good at writing DNA and proofreading to take care of the bulk of the mutation repair. All right, so again, as you prepare for class on Monday, make sure to prepare yourself for going a little bit deeper, talking about some of these mutations in more detail, and even talking about some other mutations and some other repair mechanisms.